The aim of this procedure is to use the fluorescence in situ hybridization technique to determine the sex chromosome or translocation status of in vitro human embryos at risk of genetic abnormality. This is accomplished by lysing a single cell biopsied from a day 3 embryo onto a glass slide and air drying the nucleus. The second step is to co-denature and hybridize the target nucleus DNA with DNA probes labelled with fluorochromes of different colours. Next, excess probe is removed using a stringent wash and the nucleus is stained with a DNA-specific fluorescent stain. The final step is to view the nucleus using a fluorescence microscope and to capture digital images. Ultimately, results can be obtained that show the copy number of the targeted chromosome regions through fluorescence microscopy.
Generally, individuals new to this technique will struggle because it is at the very limit of the fluorescence in situ hybridization technique. "24 hours in advance, prepare the cell lysis buffer for spreading cells. Filter the solution and dispense 1 ml aliquots into 10 to 15 1.7 ml sterile microcentrifuge tubes. Close and label the tubes prior to freezing at -20 degrees Celsius. Just before the biopsy procedure, defrost the lysis buffer at room temperature. Score a small circle, approximately 5 mm in diameter, on the underside of an amine coated slide using a diamond pen, and then, using a hard 4H pencil, pre-label the slide. Blot with a